My name is Peter Neubauer, and I'm the head of the chair of Bioprocess Engineering at the Technical University of Berlin. In the following slides, I want to present you the activities of our department. Our group consists of 6 laboratories that focus strongly on methodological development, but also apply new technologies in interesting and exciting bioprocesses. Our activities range from industrial processes in the field of circular bioeconomy to the production of sophisticated proteins in the biopharmaceutical field. We also have a biopilot plant in which we can carry out cultivations on a scale of up to 100 liters, including all downstream processing steps. A special development is our fully automated future laboratory, known as Kiwi Biolab. In this laboratory, we are pursuing the vision of fully autonomous bioprocess development. This exciting project will be presented in a separate lecture by my colleague and head of this lab, Nico Cruz-Bonasso. Over the years, our department has been associated with 3 company spin-off projects, and we have founded 2 non-profit organizations, namely the Biopat e.V., which organizes this forum, and the Bioproscale e.V., which hosts the International Bioproscale Symposium as the world's leading scientific conference on bioprocess scale-up and scale-down. The next 8th symposium will take place here in Berlin in April 2024. This slide is quite old, but shows the basic strategies of our laboratory. All our projects and considerations start by considering the characteristics of the bioprocess at the targeted industrial scale. The conditions in this process dictate the whole developmental process. Based on the technical parameters of the industrial process, we define the scale-down systems and the conditions to be used in the lab. We use sophisticated analytical methods to understand the cell physiology and, most importantly, define the scale-down criteria based on the biological properties. New types of products and cell factories are characterized using mathematical models in our high-throughput facility. These high-throughput experiments are typically used to calibrate the parameters of the model, which is challenging due to the time dynamics and the effects of history. The tight integration of parallel, well-controlled cultivations with rapid analytics is key to performing this parametrization while the process is still running, and to implement feedback control strategies with the aim to extract the best possible information from such an experiment. Finally, in process development, information from the reactor description is complemented by dynamic cellular reaction models, and even population balance models can be implemented. Over the years, our group has dealt with a variety of microorganisms in the context of scaling strategies. As inhomogeneities are a major problem on the industrial scale, our group has often used compartmented reactors and pulse-based feeding scenarios that mimic these conditions. This work on large-scale processes has also led us into the field of characteristic processes and circular bioeconomy. In terms of products, we have worked with a large number of difficult-to-express proteins, the most actual group being the hydrogenases, which are interesting in terms of hydrogen production or utilization, and they have very complex structure. Other proteins from our recent projects include nucleoside-modifying enzymes, ribosomal and non-ribosomal antimicrobial peptides, antibody fragments, elastins, and proteins with incorporated non-canonical amino acids. In the field of connected bioprocesses, we are also working on unsaturated long-chain fatty acids such as docosahexaenoic acid and on polyhydroxyalkanoates as interesting biosynthetic and fully biodegradable bioplastics. In the right-hand column, you can see some of the technologies that we have developed or that we have adapted together with our industrial partners, such as different kinds of scale-down bioreactors and in particular novel sensor systems such as the SOPAT inline microscopy and the PDW spectroscopy. Recently, we have made many exciting developments in the area of digitalization, automatic data evaluation, and modeling in connection with our Kiwi Biolab. In our Smart Bioproduction Grids group, we are interested on the flexible and efficient use of available feedstocks from the food, agricultural, and forestry industries. Our strategies include to apply in these processes modular concepts, for instance in the form of plug flow reactors, to separate the different biological reactions. We are also considering process coupling by optimizing the anaerobic process for the production of short-chain fatty acids, which are then used in a sterile process, that means single-cell process, as substrate, for instance, for the synthesis of long-chain fatty acids or bioplastics. Furthermore, we are interested in processes with two or even more organisms, so mixed cultures, and the possibilities of how to control such complex processes. In this group, we have used a wide range of stainless steel and single-use bioreactors and also a wide range of sensor technologies. Finally, if we consider biorefinery concepts and connected processes, in such projects we have also used life cycle assessment to evaluate the impact and the criteria for optimization. Here I would like to shortly go into a little bit more detail into the area of difficult to express proteins. Despite functional protein expression is a standard technology today, many proteins are still difficult to express in reasonable amounts. This is, for instance, superlarge proteins, proteins with heteromeric domains, strongly reduced proteins, proteins and peptides with post-translational modifications, proteins with overrepresented amino acids, or if non-canonical amino acids are incorporated, or even protein with cofactors. We have been successful here by not only considering genetic factors, but applying very strictly controlled cultivation conditions to keep the cell in a balanced situation. The key for this is the fed batch technology, which we use in industrial scale bioreactors, and the idea that this is applied as a kind of diet which has to be optimized to balance the cellular reactions for the synthesis of amino acids, synthesis of proteins, and protein folding. For all of these proteins, we could obtain, by the strict application of this principle, jointly with balancing of the expression, an increase of the functional amounts by orders of magnitudes. One key invention in this aspect was the N-based technology. Here we have already during my time in Finland developed a kind of a fed batch system, which is however not controlled by an external feed pump, but by an internal biocatalyst, which is catalytically applied to supply glucose from glucose or soluble glucose polymer in a controlled way. And the really breakthrough of the system is that can be applied in any scale from miniature fermentations up to industrial scale and work in a scale independent way, always in with a very good control of the conditions. This gave us the possibility to really screen, for instance, in 96 well plates, the different proteins and contracts by the use of the fed batch technology and really find optimal scenarios for their production. With this slide, I only want to illustrate at the example of hydrogenase that this facility can be then used to screen different clones or conditions fully automatically to boost the functional expression. Currently, we are still establishing the automatic strategies, but soon hope that we can use it not only for our current model enzyme, but for the optimization of other even more challenging hydrogenases. By our projects in the last years, we have learned a lot about the challenges of digitalizing all activities in a lab. In a very exciting project together with Siemens and BioNucleo, we have in an illustrative way implemented all steps from the bioinformatic part of searching for new biocatalysts over the molecular biological construction of the cell factories, the expression optimization, and the final production of the biocatalysts up to their characterization. Although we came in this project closer to our vision, there remains a lot to do to really solve the challenges of a fully automated process development. In the short presentation, I was only able to present some of our activities, but I deeply thank to all of our co-workers to really contribute to these exciting developments in bioprocess engineering. And I thank, of course, also all of our partners, the people and organizations who finance our work. And finally, I thank you for your attention.